Good morning from Buenos Aires. Good morning to our colleagues in Latin America, and good afternoon to our friends in Africa. Welcome to the virtual conference, Wet Production System and Breeding Technologies. My name is Guillermo Sanchez. I will be moderating this event. This is an activity that it was developed within the framework of a serial of international seminars led by the Argentine Fund for International Cooperation. This event is being broadcasted through the INTA YouTube channel and is also being recorded. Please, if you have some trouble, please contact us in order to help you to solve the inconvenience. So we are starting the seminar, and in the first place, I will pass the floor to the Argentine ambassador to Tunisia, Mr. Claudio Rosenweit. Mr. Ambassador, the floor is yours.
Muchas gracias. Thank you very much, and good afternoon to everybody. First of all, I would like to express my pleasure for being able to be present today in this important activity, which is a fundamental step forward in the framework of the technical cooperation project that our countries have been developing almost for the last 3 years on wet production system and breeding technologies. Also, I would like to express my gratitude to the authorities of the International Directorate of International Cooperation of our Ministry, FO.AR, to the INTA, and to their counterparts in Tunisia, as well, especially to the focal points from INAT and INRAT. I see Yusuf Fria here, and who have worked a lot in order to make some progress in this project. I also, thanks to Ambassador Mohamed Ali Ben Habib, who is a very close friend of mine, the ambassador of Tunisia in Argentina, and to Engineer Carlos Parera, representative of INTA. As you may know, our country is a traditional stakeholder in agrarian matters, in which is the result of decades of a continuous evolution of agriculture technologies developed in Argentina by INTA. This prestigious institution, Argentine prestigious institution, is a key actor of that dynamic, not only in research, but also especially in vulgarizations. One of the most significant tools that our producers have in Argentina is that they have at their disposal the INTA in order to improve their results and to overcome different kinds of setbacks. We are proud of that as a country, and we strongly believe in this way of working. We are also proud of our ability to share this know-how all over the world by the FO.AR. The FO.AR has been financing projects like this one worldwide since 1992. The different activities executed in this project were implemented and developed thanks to the support of our networking of embassies and their staff working together with the local focal points and the Argentine expert. I would also like to emphasize that the situation that we are living today in the world, because the pandemic reflects that the importance of cooperation and joint work, since there are problems and threats which cannot be solved an individual or unilateral matter. The project on wet production system and breeding technologies is an example of this success that we can reach working together through technical cooperation. It is also an evidence of the large bilateral agenda on cooperation that our countries, Tunisia and Argentina, have been developing in the last 3 years. I firmly believe that this project, among other projects, is one of the most effective ways that we have in order to improve the life of our national, whether they are Argentinian or Tunisian, through international cooperation. This is why diplomacy and foreign policy are quite important for our peoples. They really can improve the life of persons. To conclude, I would like to thank again to all the participants and wish them the greatest success in this activity, looking at the future of this project and hoping that we can meet personally again and work together as soon as possible at the end of this cauchemar, at this pandemia and this nightmare. Gracias a todos. Thank you very much as all. And thank you at the INTA for all this work. Thank you very much.
Thank you, Mr. Ambassador. Now we will listen to the Tunisian ambassador to Argentina, Mr. Mohamed Ali Ben Abid. Mr. Ambassador, the floor is yours.
Thank you, dear moderator. Good morning, or good afternoon, everyone. Excellency, dear friend Claudio Rosenberg, Ambassador of Republic of Argentina to the Republic of Tunisia, dear representative of the Director General for International Cooperation at the Ministry of External Relations, representative of the Ministry of Agriculture, Livestock and Fishes, Director of National Institute of Agriculture Technology, INTA, and representative of the National Council for Scientific and Technical Research from TSET, dear moderator, Tunisian participant, Tunisian Argentinian, good morning. Allow me first to express my sincere gratitude to the Ministry of External Relations for giving me the opportunity to say a few words at the opening of this distinguished meeting, which testifies once again the excellent friendship between Tunisia and Argentina and demonstrated the strong interest of our two countries to further strengthen bilateral cooperation. I would also like to express my deep appreciation for this very good initiative, which brings together several partners and specialists on a global topical issue, which is biotechnology applied to seeds. Argentina is considered as a pioneer in this field and has world-renowned know-how. As such, I would like to congratulate the Argentinian researchers and specialists in the field for the recent approval by the relevant Argentinian authority, it was in October 2020, of the more drought-tolerant wheat variety incorporating HB4 technology for both production and consumption. An innovative and unique agricultural technology that can minimize production losses, optimize yield, and help achieve global food security in this climate change context. Ladies and gentlemen, the agriculture sector in Tunisia plays an important role in the national economy development strategy. Indeed, the sector continues to contribute significantly with about 11% of GDP and 25% of employment in the labor force. The agriculture products account for about 8% for the economy export earnings, and the sector is increasing, attracting foreign investment. Tunisia is one of the largest agriculture producers with a share of 6% of the world production. Is the largest producer and exporter of olive oil in the Southern Mediterranean and the second largest in the world after the European Union. It's also the world's largest producer and exporter of dates. The field crop sector, mainly wheat and barley, occupies a large part of the arable land, 2 out of 5 million hectares cultivated, which demonstrates the importance of the chain of cereal cultivation in the map of agriculture production in Tunisia. Mainly based on rainfed agriculture, grain production in Tunisia remains vulnerable to climate change. Indeed, crops remain dependent on the rainfall, promoting the country to import variable amounts of wheat and barley each year to meet its needs. To remedy this situation, public and private actors work together to improve the productivity of the field crop sector and to address its vulnerability to climate factors through the search for creative and practical solutions to ensure food security and reduce maximum invoice of imports. Ladies and gentlemen, mindful of the strategic role of the agriculture sector in the country's economic growth, the government of Tunisia, as part of the 5-year Economic Development Plan 2016-2020, introduced a series of reforms aimed at providing the primary sector with high added value, greater competitiveness, and good attractiveness for investment, so as to increase its contribution to the national development effort. To achieve these objectives, Tunisia, which continues to rely on its own research, is also working to further consolidate its cooperation relation with its main partner, including, of course, Argentina, to identify new ways of exchanging experience on new cropping techniques and to seek opportunities for sharing innovative goods practices in order to strengthen national expertise in the field of agriculture. In this regard, I would like to congratulate myself on the dynamic which has marked technical cooperation between Tunisia and Argentina in recent years. And I would like to praise the good performance of the cooperation project currently underway, mainly in the field of agriculture and regional development, in the framework of the cooperation with the FO.AR, Argentina Fund for South-South and Triangular Cooperation, and in application of the terms of the Framework Agreement for Cooperation in Agriculture field concluded on 18 November 2008, and the Technical Cooperation Agreement signed on May 2019. On this point, I would like to express the appreciation of the Tunisian authority for the willingness and the Argentinian Fund for South-South Triangular Cooperation to fund 3 technical cooperation projects in the field of agriculture: biotechnology cooperation project, improvement of the bovine meat efficiency and production of sheep meat. My country wishes also to benefit from the Argentinian expertise in the farming sector, especially in the research and technical farming training fields, animal production and biotechnology, as well in the livestock sector. As such, I would like to pass on to my country's interest in diversifying this successful cooperation and to benefit from Argentina's expertise in other fields such as the biotechnology, livestock, digital economy, the vocational training, environmental protection, the renewable energy, and the civil nuclear energy, both in the context of bilateral cooperation and through the cooperation involving international bodies and plans. Ladies and gentlemen, at the end of my brief speech, I would like to reiterate to you my full disposal, as well as the staff of the embassy, to work with the Ministry of External Relations and with the technical department of Argentina, involved in promotion of scientific, technological, and economic cooperation between our 2 countries. I cannot conclude without commending the commendable effort of the management team of the international cooperation, who facilitated the realization of this meeting. Once again, thank you for the invitation. Thank you for your attention.
Thank you, Mr. Ambassador. Now I will pass the floor to Mr. Carlos Parera. He is the National Director at the National Institute of Agricultural Technology, INTA. Carlos, your time.
Good morning from Argentina. Muy buenos días desde acá, Argentina. Thanks you very much to the ambassador for both countries for participating in the opening of this seminar. I also want to give a special greeting to our colleagues from the National Agronomic Institute of Tunisia for sharing this seminar that I hope will allow us to strengthen our research and development to build competitive and sustainable agriculture, agri-food, and agro-industrial system. Also, I want to give a special greeting to our technical staff who is participating in this seminar. As the National Director of INTA, I would like to highlight that our institution has different cooperation instruments to promote our relationship with partners in Africa through agreement with public and private sector, through course, multilateral network, and by participating in research projects with funding from the Ministry of Science, Technology and Innovation, and technical cooperation projects funding by FO.AR. These tools allow us to have an active collaboration with 12 countries in Africa, specifically between 2016 and 2020 in terms of technological cooperation. We have promoted 17 FO.AR projects, executing 55 international missions that encourage the permanent interaction of more than 100 professionals from Argentina in Africa. Regarding this seminar, we hope that INTA specialists will contribute to exchange knowledge in the generation of wheat cultivars, one of the most important production worldwide given its increasing demand. Within the current context, coordinating activities aimed at sharing experience on innovation for the growth of productivity sector represent a major priority for our institution. These activities, which are at the service of our society, commit us to respond to problems in a more creative way and to support the opportunities generated by international cooperation initiatives. I hope this meeting promotes more alliance between public, private, and civil society actors to provide better solutions to global agriculture challenges. Thank you very much for your attention. Thank you.
Thank you, Carlos. Thank you to both ambassadors. And I would like to mention that is also present Ms. Alicia Barone, General Director of International Cooperation at the Foreign Affairs Ministry. She will talk to us in the closing part of this seminar. So we will start with the technical presentation. I would like to ask to the speakers to be careful of the time. Every speaker will have 10 minutes to the presentation. So let's start. After the presentation, we will have a time for question and answer. So the first presentation will be given by Fernando Salvaggiotti, who is a researcher in the plant nutrition and soil fertility and works with the crop soils and water management group at INTA Oliveros. He got a master in crop production and a PhD in soil and water science. He is also a researcher at the National Council of Scientific and Technical Research from Argentina. Fernando, your 10 minutes, please.
Okay, thank you, Guillermo. I have a brief presentation here about wheat within the crop systems of Argentina, just to figure out how wheat is managed and planted in our country. So this is Argentina, the main area of crop production is in the southern part of South America. And in the last year, in 2018, close to 6 million hectares of wheat were planted. In this area, this is mainly in temperate climates, but also some in subtropical climates in the northern part. Look at this, that wheat represented in 2018 23% of total cereal production and 14% of total grain production. Is a very important cereal for us. It's the main winter cereal for us. And if we can split the production, we can see here that most of the production and most of the area is in this temperate region. That is what we call the Pampean region, that is close to 65 to more than 70% of production is there, and more than 70% of the area. In this other area, in the northern parts, are marginal area for wheat, and the climate is more hot, and it's not so good for the crop. Just for you to have a look of how it's distributed in our country. We can see here is the share of total area with time. This is from the 60 to last year. And we see here that wheat mainly, and always also maize, have a good proportion of the area crops by crops at the very beginning. But after the 80s, soybean is the main actor in our country in the crop area. But and wheat and maize were displaced by the soybean planting. But when we analyze the same share, but take into account production, we still see that even when soybean is the main actor here, with also with maize, wheat still have a good proportion. It's the third crop in total production share in our country. And even when the area was reduced in the last year, the management and the genetics allow us to have a good proportion of the production still important. With all production is made under rainfed conditions, and we have the main soils that which are planted. They are molly soils, but also vertisols, alphisols, and entisols that they are specified by USDA. And these soils have an effective depth of from 30 to 200 centimeter depth. And that is one of the main constraints that all crops in Argentina would have, depending on how deep can these roots go deeper. Organic matter, that is the main soil health indicator, is from 2, we will see 2018, we can see here that 2 to 3% is in the northern part of the Pampean region, and we can go to 4 to 5% in the southern part of the region. And this is what implies that we have nitrogen deficiencies important for wheat. This is the soil pH. When you have a whole, our pH are not so limiting as in the tropical area, like Brazil, for example. Our pH range from 5 to 6, and there are very good pH values in soil for crop production. Phosphorus content, look at this in the right parts of the slide. You can see that our phosphorus contents are below 15 per million. That is, we have phosphorus deficiency in our soils. So these are the main chemical constraints for crop production in general. And we will see how was the last year the spread. We can see that wheat was, the red points here, was mainly planted as winter crop soybean, that's the double crop. And in the southern part of the country is mainly planted as a solely, as an only crop. So these are different management options for the crop production system. When we analyze how it represents this double crop, we see it here in the red, dark red area. We see that single soybean is the main crop in our area, but we have an important part of soybean that is planted after wheat. So wheat is in the northern part of the area is planted mainly as a double crop. And what happens? When we have the single crop soybean, we have all these years, we have only 5 months of the year occupied by a crop, but only 30% of the radiation that is in the whole season, and 30% of the rainfall is not used for crop production because we don't have any crop here. So double crop soybean in this area is an adequate option for increasing resource efficiency. So we can fill up all this time with a crop and use these resources that are not used in another way.
Fernando, so, 5 minutes.
Yeah. 5 minutes? Okay.
5 minutes, sorry.
Okay. So we have, by making double crops, we increase 20% what is the radiation interception and 30% increase in water use. Wheat is key in crop rotations. Look at this. This is in the upper part. We see how is the rotation with soil monoculture. We see how important are wet residues, and these are the same happen with maize residues. And this is important for sustainability. When we see here, we can see here that wheat make an even distribution of roots in the profiles, and that favors what is a structure of the soil and carbon content. And main rotations in the area are mainly in the Pampean regions are maize, soybean, wheat, and wheat is always at the double crop. We can include some rotations that include soybean and pea, but always as a double crop. When we go to the northern part, the subtropical parts, it's different. The rotations wheat is filling a gap between 2 soybeans, or a soybean and maize over here, or sunflower. It's not a high production for wheat. And here in the southern part is only wheat, or mainly maize, soybean rotated with maize and soybean, or only that's a double crop. And we go to more drastic rainfall conditions. Wheat is planted after 2 years. So we have a fallow wheat, 1 year of fallow and wheat. So concluding, Wheat is central, and I try to make this in this presentation, I hope so. Wheat is central for soil restoring and maintaining soil fertility in the cropping system of Argentina. This is the amount of residue that lives in the soil is important for making good carbon balances, and that's contributing to sustainability. And wheat is an important option for increasing resource use efficiency at the system level. I show you that how we improve use of water and radiation if we use double crops when possible. What are the limiting factors for production in our systems? Are mainly water availability, depends on the region, but this water availability depends on the rainfall pattern of in each region, the soil holding capacity of the soils and the depth of the soils, the presence of shallow groundwater. I didn't have time to talk about this, but we have regions in which shallow groundwater may be a problem. We have nutrient deficiency, nitrogen, phosphorus, sulfur deficiencies, and zinc deficiency that we had to deal with. We have a limited factor in soil quality sometimes because of the soil monoculture, or as all the crops we have some issues with weed, pests, and diseases. What are the challenges in crop production with our systems? First of all, try to reduce our yield gaps. We have yield gaps that we have to reduce with management or genetics to improve our soil quality, and where wheat is a central actor for this. We have some spatial variability in crop production in some areas that we had to deal with. And I think that one of the great challenges in our system is to manage nitrogen fertilization, manage nutrition for increasing both yield and grain quality in terms of protein. Well, thank you very much. And I hope there are some questions from the public.
Thank you, Fernando. Thank you for your very, very good presentation, and excellent presentation, and excellent with the time. So we are going to the next presentation. That is about, sorry. The next presentation is about wet crop physiology for climate change adaptation, and it will be presented by Fernanda González. Fernanda is agronomist, and she has a PhD in agronomy sciences with a special interest in crop physiology. Dr. González works in INTA Pergamino, and she is also a member of the National Council for Scientific and Technological Research. Since 2009, she is professor at the School of Agronomy, Natural and Environmental Sciences at the Northeast Buenos Aires Province National University. So, Fernanda, you have your 10 minutes.
Well, thank you, Guillermo, for the presentation, and we'll try to show you the main research lines we are working on to cope with climate change adaptation. And well, we all know that climate variability represents a risk for crop productivity, and that climate change increases this risk. For us, climate change began during the second half of past century, and for our humid and central zones, that would be these areas, we know that it meant an increase in average temperature, mainly associated with the minimum temperatures, a decrease in the days with frost, an increase in the total rain and extreme events, and particularly in this central zone, an increase in the dry period during the winter. And we know that in the near future, temperature is going to keep increasing, no more than 1 grade centigrade. We will have an increase in the heat waves and extreme temperatures. We will have no change in rain or a slight increase, and we will have more extreme rain events and higher waterlogging risk. And also this dry period in this central area will increase during the winter. So we know that these changes have already modified our environment. And if you see the yield, this is the simulation of yield potential for wheat in Pergamino, that is located here in the north of Buenos Aires province. from 1930 to 2000, and you can see that the yield potential has decreased, steady decrease. And if we focus in the last period from 1970 onwards, this decrease was even higher. And this is only a consequence of the increase in the average temperature during the growth cycle. We know that, in fact, the most important thing is the increase in the minimum temperature during October and November. That is when the crop is during stem elongation, anthesis, and grain filling periods. And more recently, it was also simulated the decrease in yield potential for a bigger area. And the conclusion was that we already have from 2 to 7% decrease in yield potential for each grade centigrade increase in minimum temperature. So the environment has already changed, but despite this impact of climate change, we have a steady increase in the yield at farm level. This is the yield of wheat at farm level, and it increased at 35 kilograms per hectare per year. And partially this was a consequence of the increment of the yield potential of the cultivars. Here we have the yield potential of the cultivars. These are 10 cultivars released during the last 50 years. It was measured in Pergamino, and we have an increase of 42 kilograms per hectare per year. And we know that this yield potential is expressed under a wide range of environmental indexes. Here we have the yield and the environmental index, and we can see for different growth cycles, we can see that the modern cultivars outyield the older ones in a wide range of environments, except for these environments of 2 or 3 tons per hectare. So we think that it is interesting to keep on increasing yield potential. And if we think about that, we are focused on spike fruiting efficiency as a potential trait to increase it. We know that the grains per spikes depends on the spike dry weight at anthesis and the fruiting efficiency. That could be the number of grains set per gram of spike dry weight. And we know we can measure it if we use as a proxy, if we use the chaff of the spike as a surrogate to a spike dry weight. And from the more physiological perspective, we know it will depend on the fertile floret efficiency. That would be the number of fertile florets that are set per gram of a spike dry weight, and on the grain set. We already show that we can use the fruiting efficiency at maturity as a phenotypic selection criteria to increase grain number and yield potential. And we already know the ideotype of the spike for high fruiting efficiency. And recently we identified and validated 2 QTLs for these 2 traits that can allow us to increase from 5 to 3% the yield of the spike. So this is extremely interesting for us, and well, together with Leonardo Vancetti, we are trying to identify candidate genes in these areas. Another trait we are interested in is the photoperiod sensitivity. For this, we are working with different isogenic lines that combine different alleles for PPD1, and we're trying to quantify the response to photoperiod. For example, here we have the thermal time from emergence to anthesis in relationship to the photoperiod, and we can see how different combinations yield different photoperiod sensitivity. And we are interested not only in the time to anthesis, but also in the different phenotypes of wheat, for example, from emergence to beginning of stem elongation, or from beginning of stem elongation to anthesis, just to cope with and to counterbalance the increment of the minimum temperature I mentioned earlier. Finally, if we think in the water, in the yield that is limited by water, this is the variability we have. This is the climatic variability and the probability of drought in Argentina, and this is the probability of waterlogging. These are 2 different sowing dates, and you can see that we have areas with high risk or high probability of drought in Argentina, mostly in the west area or in the central area, and there are some places where we have some waterlogging problems. If we see the attainable yield in this area, you could see here, for example, the blue is a higher yield. It's from 5 to 6 tons, and this is less than 1 ton. And you could see that more or less the attainable yield follows this drought pattern. And we know that in the near future, we will have simulations predict a reduction, an average reduction of 13% in our yield, independently of the CO2 scenario. And this will be a concentrated reduction here in this area that that Fernando mentioned previously, that has a high risk of drought. And just to cope with this, we are working in a cooperative program with
I'm sorry,
partners.
Fernanda, 5 minutes.
Okay, with different partners from Brazil, Paraguay, Uruguay, and Chile, and some universities within the cooperative program for technological development in agri-food and agro-industry in the southern corner of America. And within this project, we have the objectives of the characterization of regional germplasm to biotic and abiotic stresses, the identification of physiological and genetic bases associated with yield and stability under such stresses, the development of a modern high-throughput phenotypic platform validated at regional level, and agronomic simulation models to quantify current and future abiotic stresses according to different management strategies. So we are working in multidisciplinary teams focusing on phenotyping, genotyping, and modeling, particularly for the identification of physiological and genetic bases of wheat tolerance to drought. Right now, we are running multi-environment trials with cultivars with different drought tolerance in different locations that cover a wide range of latitude and longitude. In these experiments, we are trying to validate the phenotyping platform. So we're trying to correlate the traits we will measure with image taken by drones. And particularly for the drought tolerance, we have experiments in Chile and Argentina in 2 locations where we can control the water supply because it is the Atacama desert, and the irrigation valley in San Juan. This is very, very dry, extremely dry areas. And where we think we will have a good results here, just to try to identify some QTLs interesting for improving the drought tolerance. Well, this is all I wanted to share with you, and I am available for questions.
Thank you, Fernanda. Very good presentation, very clear. Thank you. So now the next presentation will be in charge to Marcelo Helguera, who is going to present INTA's wet breeding program. Dr. Helguera has been working in INTA since 1992. He obtained a PhD degree in biological sciences at the Córdoba National University. He got a position to start the project of biotechnology laboratory in INTA Marcos Juárez, linked to the wet breeding program. So, Marcelo, you have your 10 minutes.
Okay. Okay, thank you very much for the presentation, Guillermo. Well, I will give you a quite short presentation about what we are dealing in the international wheat breeding program. Well, some historical milestones. We can say that the institution where we are working, which is the National Institute of Agricultural Technology, was founded in 1956. Three years later, the experimental station Marcos Juárez was appointed as a headquarters of the wheat breeding program. Since this year to now, more than 100 India wheat cultivars have been released, which shows the wheat breeding program as a successful program. From the wheat breeding program, the main goal is the release of wheat cultivars with high yield, broad adaptation, and good bread making quality. Some of the main constraints that we have in the crop are disease resistant, diseases like leaf rust, yellow rust, stripe rust, Fusarium head blight, and Septoria tritici. Constraints, well, some of them have been described in previous presentations. We can talk about the drought as a very important limitant, and also extreme temperatures. Other thing that we consider a main constraint is the adaptation to no tillage conditions, which are one of the main management systems of wheat in Argentina. Some of this was very well explained by Fernando in previous presentations. Okay, let's go on. Well, here I wanted to show you the current location of the INTA wheat breeding program. Here you can see on the left of the presentation a map of Argentina, where you can see the central and north part of the country. Here in green, you can see the region where wheat is planted. And inside of the green region, what we can see are different dots in different colors. The red dots that we can see here belong to the major locations of our wheat breeding programs, where most of the activities that we do there are breeding and evaluation. Some of them, going from north to south, are Paraná. Here is the northest point, red point. Then we have Marcos Juárez, which are the headquarters of the program. Then we have Pergamino, and then we go to the south to Bordenave, Balcarce, and Barrow. Then another group of dots that we have in the map are the blue ones that belongs to the annex fields of evaluation. Here we have the northest one is here and belongs to 3 locations that are very close each other, which are Corral de Bustos, Monte Weiss, and Los Molinos. As far as we go to the south, we can see San Antonio de Areco and Bragado, and in the southeast region, we can see we are very close to the Atlantic Ocean, Miramar. The main goal of this annex field is the evaluation of the advanced breeding lines of the program. And finally, we have a third kind of locations, which is the disease nursery, in order to characterize the germplasm of the wheat breeding program, the performance of this germplasm against the main diseases. And it's here at the northeast of the country, in a city known as Reconquista. And well, then other thing that I wanted to show you are the selection and evaluation activities of the wheat breeding program. As you can see here, most main activities are breeding and selection of population of the program. Then we have the evaluation of advanced breeding lines, and a third very important activity of the program is the seed increase and the maintenance of purity of the varieties of the program. Other thing that you can see here is that most of these activities are in red. That means that the 6 major locations that are included in this program, they deal with these 3 kind of activities. That is the management, the breeding population of the program that come from F1 to F7. Then something that is done in 2 of them, which are Marcos Juárez at the north and Barrow at the south, is the crosses. That is the starting point of the material. We do around 350 crosses in Marcos Juárez, and maybe a number similar in Barrow, maybe not so much. And here the number that I'm showing you here of populations that are considered in the program are the number from Marcos Juárez. In general number, we can say that every year we handle around 550 plots for breeding and selection. In Marco Juarez, similar numbers are also managed in another major location. In the case of evaluation, the number of plots that we manage every year is a little bit more, maybe 6,600 plots that give us around 11,000 plots every year in one location. If we multiply by 6, we get around 50,000 plots every year managed in the wheat breeding program of INTA. Here, well, also other thing that you can see is that the red dots in this part of the activities of the program, which is the evaluation of advanced lines, that means that the annex field take part of the analysis of preliminary multi-location trials where the best characterized materials are taking part. Also, the regional yield trial, where we get every year the information of the advanced lines from the breeding program, and the best one will be released as new cultivars. This is
sorry, Marcelo, 5 minutes.
Yeah, 5 minutes, okay. The disease nursery, what it does is the evaluation of the regional yield trials. The material here are also analyze the behavior against main diseases, and this information is obtained by this location. Okay, then what? How do we deal with quality in the breeding program? Well, what we do basically in the early generation of the breeding program, what we do is a first selection for quality, considering gluten strength, using tests like Seleni tests that they need not very much amount of seed for that. As far as we go in advanced generation of the breeding program, we can have more amount of seed available, and we can and obviously we include a higher amount of tests for a better evaluation of the material for bread making quality, gluten performance, and also the quality of the seeds. Finally, here we can see in the regional yield trials that we have a very good numbers of different tests for a better evaluation of the performance against bread making quality of the materials of the program. And with the information of all these tests, we get something like is a bread a quality index, and according to the information that we get in this index, the future cultivars will be classified for quality as quality 1 with extra strong gluten. These are the material with highest protein content and strongest gluten strength. And as far as the gluten becomes weaker and the protein content goes down, we get traditional baking and direct baking. This is more or less to give you a very preliminary idea. Related with biotechnology, what we do is first we do the characterization of the crossing block of the breeding program using markers for major traits for the breeding program, like it could be bread making quality, disease resistant genes, and also the adaptation genes like photoperiod response and vernalization response. Based according to this, the breeders use this information in order to make smart crosses every year, which is the starting point of the program. Other thing that we do is mutagenesis in order to get valuable and new ability useful for the breeding program. An example that I bring to the presentation is the identification of novel high molecular weight glutenants, which are storage proteins valuable for the determination of bread making quality that could be useful for the breeding program. Another thing that we do is marker acetate selection as a strategy to include or introduce new sources of variation valuable for the program, like for example, disease-resistant genes like LR47 that was introduced in the first cultivar released in Argentina with this technology, with this INTA 2004. Then Another example, more recent, is the LR47F and FHB1, two disease-resistant genes introgresses in the cultivar MS INTA 416 that was released in 2015. And finally, what I wanted to show you is the team behind all the activities that I'm showing you in very short period. But the idea that I wanted to give you is that we're an interdisciplinary group. Here in different color, we can see the disciplines of researchers involved behind the release of new cultivars from the program. The researchers in black are people working on breeding. Here in green, we can see the people that are phytopathologists working in the disease resistance of the breeding program. In blue, we have the biotechnologists. In red, we have the ecophysiologists of the team. And finally, in orange, we have the researchers focused on bread making quality. Well, this is all I wanted to show you in the presentation. I really hope that you find it interesting, and if you have questions, I will be pleased to answer them.
Thank you, Marcelo. Very excellent presentation. Very good. So the following speaker is Leonardo Vancetti, a specialist in genetics who completed his doctoral study at the National University of Córdoba with an orientation in plant breeding. He has been working at INTA since 2004 in the Biotechnology and Genetic Resources group at INTA Marcos Juárez, and he is also researcher at the National Council for Scientific and Technological Research. Leonardo, you have 10 minutes, please.
Thank you very much for the presentation. I'm going to talk about new technologies applied to wheat breeding. Sorry for my English. I'm going to talk why our breeding program is moving from conventional markers to KSP markers. Also going to talk about two platforms that we are using to move faster in our breeding program, is specifically a speed breeding platform and doublet haploid platform. Also going to tell you about two applications of genomics in our breeding program. One of them is the exon capture, and the another one is the use of machine learning for genomic prediction. Also, we're going to highlight a positive aspect of this application and not so good aspect to improve. Our breeding program is using molecular marker for almost 20 years. Marcelo is one of the pioneers of this technology in our breeding program. But recently, a lot of high throughput genotyping platforms have been development, and the KSP molecular markers are a good option to improve our breeding program. The conventional molecular markers, like I show you in the left of the presentation, is take a long time to run, around 6 hours to get a good results, including the procedure include the amplification of the DNA, restriction enzymes, and agarose gel electrophoresis. And the KSP marker take only 1.5 hours to get a good results, and are a good marker for our breeding program. Has a positive aspect. The KSP marker are codominants, has lower cost, and are faster than the conventional molecular markers. You can transform the genotyping, high throughput genotyping platform SNPs into individual PCR markers. You don't have contact with acrylamide or ethidium bromide, and are scalable in processivity. yes, has no positive aspect. We need to import the master mix for KSP, and in Argentina it's a problem. And you need a real-time PCR machine equipment to run this kind of molecular markers. But I think we believe that the use of KSP will help us a lot to make faster and better characterization of our genetic our germ plasm, and also to get a better selection for interesting genes. Okay, about our speed breeding platform. The speed breeding methodology is increasing usage in breeding program, and is based in placing the plants under controlled conditions of light and temperature. Yes. Recently, the conditions for wheat have been published, and we replicate these conditions in our breeding program with some modifications because we are interested in to maintain the winter genotypes, and the condition of speed breeding is very fast, as some winter genotypes don't spike and don't get sick with that genotypes. Because that we introduce an additional step of vernalization in the platform, but we can get in our platform 4 or more generations per year in a reduced space. The platform also get the possibility of apply marker-assisted selection. Also, you can make an inoculation of pathogens and reproducing stress in the platform, and has no so positive aspect. You need a medium or high infrastructure. Has high operating cost, and you need training and dedicated human resources to use the platform. Yes, at 100% of capacity, we can produce 2,500 with F6 lines per year. It's almost the same amount that the conventional wheat breeding at the field. Yes, in 1 year we have a lot of plant, and this is a more platform. We can make bigger, and we hope so get more capacity in the future. Another platform that we are starting to use is not so new technology, the double haploid technology, but in INTA's breeding program is starting to use for breeding. And in general, the pathway is you emasculate the wheat, you make a pollination with maize. In this process, you duplicate the genetic content. After that, you make an embryo culture, plant rutification, grow the plant in the chamber, and you get the seed of the double haploid plants. It's very complex, but we think using both platforms, we can allow to increase our genetic gain by accelerating the times of conventional breeding. Some positive aspect of the use of this platform is you have complete homozygosity in 1 generation. You can identify recessive genes easily. You also have the possibility to apply marker-assisted selection, and has no so positive aspect are similar to the speed breeding platform. You need to high infrastructure, high operating cost, and you need training and dedicated human resource.
Sorry, Leonardo, 5 minutes.
Yes. About the genomics applied to wheat breeding, I will show you a very interesting how we say, interesting technology is the exon capture. The wheat genome is very big and very repetitive, and is very expensive to sequence all the genomes. In that reason, you can there is a technology you can extract all the coding region of the gene and sequence them in that aspect. You can get all the mutation in all the genes in easy way, and it's cheaper. Some positive aspect of the exon capture is very informative, very useful for breeding, but has negative aspect. You need some bioinformatic skill to get the genome browsed with all the sequence of the genes. But for example, it's very fast to find mutants or mutation for interesting agronomic trait. This gene GNAI1 is related with fertile flowers per spike, and this is in red is the mutation associated with that trait. You can find this mutation in some population, and after that you can easily check the effect in local environments. Here we show the effect of the mutation in two environments, in Pergaminos and Marco Juarez. Fernanda is working in that project. Also, the exon capture helped a lot in fine mapping project. If you have exon capture in the parental lines, you can get all the mutations in the region of the QTL. This QTL is, Fernanda talked about earlier, related with futin efficiency in the chromosome 5A. In this region, we have around 1,000 mutations that would be useful for developing new molecular markers, and also for search of candidate genes responsible for that QTL. And finally, another good technology I think we can use in the future is the machine learning to make genomic prediction. This is an example. We use machine learning in a small set of 100 cultivars in Argentina. We took 4 high futin efficiency and 4 low futin efficiency cultivars, and with the rest of the panel, we make a training panel for the algorithm of machine learning. And after that, we put the 8 unknown genotypes in the model, and the machine learning will separate the high fertility and low fertility genotypes only with the genetic information we give. Yes. Some positive aspect of genomic prediction. You can predict complex traits using molecular data, possibility of selecting genotypes without conducting field experiment, and also positive aspects. You require a big data set. Our data set is very small. We need to get a better data set. And there is no software or packages available to easy use this technology. Thank you.
Thank you, Leonardo. Thank you so much for your excellent presentation. Now is the moment of Ezequiel Bocio, a specialist in biotechnology with a PhD in applied sciences. He works at INTA as a researcher on plant genetic transformation. He also teaches genetic engineering and biotechnology at the Morón University. Ezequiel, you have 10 minutes, please. Thank you.
Okay. Thank you for the presentation. First, I would like to say hello to Sausan. Sausan was all the time with us when we were in Túnez. and dedicate a lot of her personal time to us. So thank you for that. And on my presentations, I will talk about the method we use for genome modification in wheat. Okay. In the lab of genetic modification or genetic transformation of crop, we work in different crops like wheat, corn, and cotton, using both methods to DNA delivery, like biolistic and biological methods for genome modifications. But in these presentations, I would like to show you our capabilities in genome modification of wheat, especially in genetic transformation and gene editing. For wheat transformation, we use the direct somatic embryogenesis method for plant recovery for in vitro culture, and we use the macroprojective environment as a method for DNA delivery. We work with two different cultivars for in vitro culture, the Bodway cultivar that came from the CIMIX, and the Proyecto Federal, that is a local variety from Argentina. We usually have a transformation efficiencies of around 10%, and this efficiency depending mainly on the size of the vector that we use for transformations. We have applied this protocol for the protocol of obtaining transgenic plant for different objectives, and including the constitutive or inducible expressions of heterologous genes, as well as for gene function validation. Now I will show you some examples of these applications. In this example, it is an example of a constitutive expression of heterologous genes. In this work, we have expressed antifungal proteins as a biotechnological strategy to improve the plant's response to fungal disease. This other work in this development is an example of inducible expressions of heterologous genes. In this work, we evaluate the expression of IPG genes under the control of a senescence inducible promoter as a strategy to improve drug tolerance. These transgenic plants were evaluated in different conditions as a growth chamber of a biosafety greenhouse, and for several years in field trials. Finally, here I will show you an example of transgenesis used for gene validations, gene function validation, gene validations. And in this work, we use vectors designed to induce post-transcriptional gene silencing as a transgenic approach or transgenic strategies to suppress the glutathione biosynthesis pathway in wheat too. Respect to gene editing, in the last 2 years, we have incorporated these new biotechnological tools for genome modifications. As you know, I think you know, through these techniques, it is possible to generate in a high accuracy way different types of modifications in the genomes, like gene knockout, gene replacements, base editings, or gene regulations. So far, we have been working just in developing of knockout genes. So for knockout gene, we are using the strategy of modular assembled vector that was published by Boitas, I don't know, 5 years ago. In this strategy, we obtain a single vector for transformations. This vector containing all the molecular genes that is needed for editing. I mean, the Cas9 enzyme or the gene that coding for the Cas9 enzymes. A cassette with selection markers for in vitro culture. In our protocol, we use the PAR genes that confer tolerance to phosphoinositidines as a selection marker. And then we have here the tandem of RNA-wise that work with the Cas9 enzymes. The vector that we obtained for these methods are big vectors, very large, of around 10,000 base pairs. This work was conducted in collaboration with the Biological Research Institute here for INTA and the Department of Soils and Crop Science in Colorado University. So with this vector, we work in the same way that we work in transformation protocols. I mean, we use the same in vitro culture strategies with direct somatic embryogenesis. And we use the gene gun for DNA delivery. And well, with this protocol and this vector, we recover in the last assays a set of plants. And after the molecular analysis by PCR and DNA sequencing, we found that these kinds of deletions, or as a gene editing, here we can see in some plants the deletion when we compare with the ball white cultivar, and here there is other deletions in this line of sequences. Other thing, or interesting thing for us that we realized in this protocol is our transformation efficiencies was very, very low, 1.35%, compared with the around 9% with the control vector. That is our expected transformation efficiency with the protocol that we handle. We think it is due to the size of the vector for editing. So in order to improve this efficiency in the regeneration of the edited plant, with the idea of work with a smaller vector for editing, we decided to obtain transgenic wheat plant that with constitutive expression of the Cas9 enzymes and marker-free. For this work, we done a co-transformation. We do two different vectors. One of the vectors containing only the gene coding to the Cas9 enzyme, other separated vector that is the in vitro selection marker. This strategy allow us later for segregation eliminate the marker for in vitro cultures. So with these two vectors, we done 6 independent assays of transformations where we bombarded around 2,000 scutellum and recover after 3 or 4 months of in vitro culture, 39 transgenic plants not express Cas9. Our idea is with this plant is made retransformations, and in this second transformation, we will work with little vectors that containing only the target-specific RNA guides, and this is a vector of 3.5 kbps, so it's a little vector and easy to transform. We expected with this, when we work with this vector, a high transformation efficiencies and a very good rate of edited plant recover. So to finish, say that with genetic transformations protocol, we can express heterologous genes or overexpress endogenous genes, as well as we can do gene validations through RNA silencings. Through the protocol of gene editing, we can do gene knockout, and both genomic modification method are very interesting tools for crop breeding as well as for gene discovery or gene validations. Thank you for your attention.
Thank you. Thank you so much, Ezequiel, for your very clear and excellent presentation. Before to continue, I would like to correct myself, because when I presented to Fernanda, Fernanda is a professor at the Northwest Buenos Aires Province National University. That's a correction. So I would like to mention that we are receiving questions for the speakers through the YouTube chat. So next, we are going to the last presentation by Agustina Willem. She's a specialist in science and technology management from the National Directorate of Bioeconomy of our Ministry of Agricultural, Livestock and Fisheries from Argentina. So, Agustina, you have 10 minutes, please. Agustina, have you some problem with the presentation? Okay, I think that Agustina is not here. So while we are waiting for Agustina, I would like to make the first two questions to Fernando.
Fine.
Fernando, we have two questions to start with the question part of the seminar. The first one, how is the wheat nitrogenous fertilizations management?
Okay, this is something crucial in our region, because we elaborate something using the content of nitrates in the soil up to 60 centimeter depth, and to an expected yield to fill out with enough fertilizer. This gives us a threshold that is from 130 to 160 kilograms per hectare, summing up both sources. But the great challenge we have is how to manage the protein content, because when we have more nitrogen in the soil, we have more yield, and we have to deal with the protein. Sometimes the good years, we have no good protein content in the grain. So this is the way. But as in our country, the farmers gets the money from the yield and not from the protein, the management is based only on yield.
Okay, thank you. We are the second question for you, Fernando. Which are the advantages or disadvantages of shallow groundwater in Argentina?
Yes, I didn't have time to show where the shallow groundwater are, but we have a plain area in our country, and when this shallow groundwater is between 1.5 to 3 meters, we have a very good advantage of this water, because the crop is like being irrigated. But when the water is below 3 meters or above 1.5 meters, you have problems of water scarcity or waterlogging. So these are how to manage. And sometimes when we have high rainfall, we have waterlogging, for example.
Okay, thank you. Thank you, Fernando. Now I have a question for Leonardo. Leonardo, the data on the various breeding technologies mentioned has not so positive effects related to ONIL, to cost of infrastructure and human capital. Could you please throw more light on this?
Yes, here in Argentina, the infrastructure is very expensive. When you work with split breeding or double haploid platform, you need high infrastructure. You need air condition, light, and for us, at least in Argentina, this is expensive. And related to human resources, also, I think the both platforms are very demand on human hours and controlling the plants. Everything is going well in the greenhouse or in the split breeding platform. We have some problem with human resource, and I think it's very important for start this kind of project, you have a very dedicated and trained human resource to produce the both platforms. Yes.
Okay, thank you, Leonardo. So I have already presented to Agustina. I think that Agustina is connected now. So Agustina, you can start with your presentation.
Okay, Guillermo, thank you very much.
You're welcome. Go ahead.
You can see my presentation. Okay. Hello, everybody, everyone. Thank you, Guillermo, for the presentation. I will talk about the Argentina regulatory framework for new breeding technologies for agriculture use. First of all, it is important to highlight the experience of the Argentina regulatory system. Here we have the most relevant milestones of the National Advisory Commission on Agriculture Biotechnology in the last 29 years. In 1991, the Commission was created and the first authorization for field trials with GMO crops were granted. In 1996, the first commercial approval of GM soy crop and the first constant seasons permission for cottons and maize were authorized. The first animal field trial was carried out in 2005. In 2011, the regulation for field trials, commercial release, and stack events were updated. One of the most important milestones of CONAVIA was the recognized as a reference centers in biosafety for GMOs by FAO in 2014. Also, events with new phenotypes we are evaluated in the same years. And in 2015, the MBT's framework became official in Argentina as the first country to develop gene editing regulation. So far, Argentina has 60 GM crops of the following species: cotton, soybean, alfalfa, corn, potato, sunflower, and wheat in the market. And we also made a new regulation update. When the MBTs began developed globally, Argentina began to look with the interest at what our peers, such as the United States, Canada, and the European Union, was developing. So CONAVIA decided to work in this issue. The Commission also analyzed the interest in research and development of the genome editing products at the national and international level. This is how CONAVIA decided to accompany these emerging technologies with an appropriate regulatory system. In order to understand the evolution of the regulatory framework for MBTs, which was accompanied these technologies of the product, we can observe the following technology. In 2016, as mentioned before, CONAVIA looks what happened with MBTs in the world. In 2015, the first regulation on MBT-only plants was officially published. In in 2019, the resolution of animal, MBT, and microorganism was officially published. And in 2020, the Biosafety Commission the Commission decides to work in a single regulation for MBTs that include plants, animals, and microorganisms. Bueno, the Commission, in the moment that the Commission decides to generate a normative of MBT, decides to use the definition of genetically modified organism that the same in the Cartagena Protocol, and define a genetically modified organism that has a new combination of genetic material obtained through the application of modern biotechnology. During the this definition is the same as the Cartagena Protocol. The draft resolution has a guide that has the different models, the general models, the organism models, and molecular model and phenotype models. Sorry, but I have a problem with my. What is the molecular modular characteristic? In the moment that the applicant can consult to the Commission if the regulatory status of the product, it is important to detail of the technique used and all steps applied and the intermediate stage to obtain the product, molecular description of the target nucleotide sequences of the organism and their state prior to apply the techniques, function of the sequences in their state prior to apply the techniques, molecular characterization of the target sequences after apply the techniques, genotypes obtained, expected obtained change in the function of the sequences and the phenotype after applying the techniques, map of any genetic construct of nucleotide acid fragment used to obtain in the process, detailing the genetic elements if applicable, analyze to possibly affect sequences outside the target sequences, and the last one, sorry, but I have evidence showing that the product obtained does not present new combination of genetic material. Here we have the remarked features that the draft resolution. The draft resolution, it is a procedure to determine if a product obtained using MBTs would be under the GMO regulation or not. The analysis is made case by case. This resolution is not restricted to a list of MBTs. If the product is still on the design stage, the developer can consult the committee about its regulatory status. The Biosafety Commission must perform the assessment in 20 working days in all cases. Use the definition of GMOs from the Cartagena Protocol, and the last one, specific annexes for animals, microorganisms, and vegetal. Here we have the procedure to determine the regulatory status of the product, and if the product does not contain a new combination of genetic material in the genome generated by MBTs, the product is not considered a GMO. Here we have some insight on the experience starting in 2015. It is possible to predict the cost and times of the products in the design stage. This regulation allows developers to get their product to the market faster. The speed of innovation of the product obtained by MBTs is higher in relation to GMOs. The proportion of techniques used is in gene editing is the 18.6%, and in others MBTs is the 14%. Relation of the consultations, the PYMEs represent the 32%. The public institutions represent the 59%, and multinational companies represented 9%. To conclude, the crops obtained by gene editing have a potential to comfort the challenges facing agricultural production, helping to increase the supply of food and other agricultural products in a sustainable manner. Considering Argentina's 5 years of experience applying these criteria in case analysis of product derived from MBTs, we can conclude that taking the definition of GMOs from the Cartagena Protocol is adequate and sufficient. The essence of the draft regulation is to contemplate all the organisms under the same resolution, independent without being likely to the commercial regulation for GMOs. This is all. Sorry for the problems with my computer. Thank you.
Thank you so much, Agustina. So we have made a screening of several aspects of wheat production, soil and water management, biotechnology, gene edition, regulatory aspect. So I would like to ask to our colleague from Tunisia that are in the Zoom platform if they want to make some comment, some question, or some observation.
Hi, hello.
Hello, Yusuf.
Hi, everybody. Thank you very much for the invitation for this kind of meeting, so we can see each other and get the links together again. First of all, I really would like to thank the Excellencies, Mr. Claudio, the Ambassador of Argentina and Tunisia, and Mr. Ben Abid, the Ambassador of Tunisia in Argentina, because it's really very important for all of us, I think, to have such support, political support, to try to find ways to cooperate. And I really would like to thank them because they were with us for all this very scientific and high-level scientific meeting. Now, I would like to thank all the researchers for these excellent presentations that gives us a very clear idea about what is going on in INTA and more globally in the area of genetic improvement and genetics in wheat in Argentina. Actually, as you might all know, we already had the pleasure to have Mr. Ezequiel and at that time Anna Clara to visit us in Tunisia, and that was an occasion to talk together about what we are doing in some of the labs in the research labs in Tunisia, and what were the possible or potential areas where we can cooperate and collaborate for a common project that might be within this triangular cooperation. As for wheat, and His Excellency Mr. Ben Abid has already introduced the importance of wheat in Tunisia, in Tunisian agriculture and the Tunisian diet. We are one of the biggest consumers of wheat in the world. Cereal production is very important also in Tunisia. It covers about 1/3 of the lands, arable lands. We are producing about 50% of our production, of our consumption of durum wheat. But as for bread wheat, we are importing about 19 to 95% of our needs. And I know you all have been working and talking mainly about bread wheat. But what we can do and what we can say also is that, of course, durum wheat and wheat in general and cereals in general are mainly rain-fed in Tunisia. We have only 100,000 hectares that are irrigated. So drought and drought tolerance is really one of the main topics and traits that we are working on in our lab, but also in various labs in Tunisia that are involved in the research and genetic improvement of wheat. Actually, we are also already hit by the climate change, and I would say that even in the best areas in Tunisia, at some time of the year, the crops are experiencing drought, and so we are working a lot, as I said, about this topic, particularly in our lab. Tunisia is really maybe one of the best places to work on climate change, because we can have, and we do have, experimental stations that are located from 150 millimeter per year up to 1,000 millimeter per year, which in a quite small country as compared to Argentina. And we already have a phenotyping platform in these different experimental stations. We have also expertise with nitrogen use efficiency studies. We do quite a lot of pre-breeding in cooperation with our institutes in Tunisia. So we do some pre-breeding for them. And I'm quite disappointed that actually the INRAT was not with us today. We really hope that they will be there, but maybe one of those days we can have another meeting so we can present some of what's being done in Tunisia in terms of genetic improvement for wheat and other cereal crops. Tunisia is also as a secondary site for diversification for barley and durum wheat. So of course, we, as I said, we do a lot of large germplasm phenotyping in our experimental stations to look for traits related to heat tolerance during flowering, and of course drought tolerance. And we have a lot of ongoing work also on salinity tolerance, because for some of the irrigated cereals, this is a very important issue for us. Yes, this is some of the thoughts that I wanted to share with you. What we have done so far as regard to our cooperation was and it's maybe the time to thank really, of course, the Ministry of Foreign Affairs of Tunisia, and also the Ministry of Agriculture. We have established and shared now memorandum of understanding with our counterpart in Argentina, and we hope that it will be signed soon, so we can go and move forward and try to have a cooperative project together in the field, and we will do, of course, our job of bringing the right persons with you from the different institutions that are involved in genetic improvement. And thank you.
Thank you, Youssef. So, Mr. Ambassador, you have muted your mic. Mr. Ambassador, you are muted.
Yes, I'm sorry.
Okay, thank you.
Thank you, dear moderator. Just allow me, once again, to thank all the participants for their excellent presentation. But I think it will be more efficient if we can. Such meeting is very interesting, of course. It's very technical. Just for me, it's very technical. What I suggest, if you can, just after this pandemic of coronavirus, if we can have a meeting in Tunis with the representative of INTA and with their colleagues from Tunisia, from the Ministry of Agriculture, and to can discuss more efficiently these topics. And this it will be very interesting. From here, I just want to call the representative of INTA through the Ministry of Foreign Affairs, of course, if we can arrange a meeting in Tunisia just after the pandemic. And this is really very interesting. Thank you very much.
Okay, thank you, Mr. Ambassador. Yes, the coronavirus will put to all of us around the world in a challenge. So I think that we are working very good in this kind of activity, of virtual activity. Well, we are passing to the closing part of this seminar. So for the closing, I will pass the floor to Mr. Martin Alous, Secretary of Food, Bioeconomy and Regional Development from the Ministry of Agriculture, Livestock and Fishery of Argentina. Mr. Secretary, you have the word.
Thank you. Hello, everybody. I will be short in time. First, I bring greetings for our minister, Luis Basterra. I would like to introduce myself too. I am Marcelo Alous, Secretary of Food, Bioeconomy and Regional Development of the Ministry of Agriculture, Livestock and Fisheries from Argentina. I'm glad to be in this meeting about wheat production and breeding because we think that all technology, including the biotechnologic ones like MBT, Agustina spoke about this, I think, are essential to face the global challenge of increasing production to feed the growing world population and also protect the biosafety of the environment. As you have already been told today, Argentina has very strong and diverse capabilities and already set up a framework to regulate GMO and products derived from new breeding technologies. And that is why we are now a pioneer country in the adoption and regulation of these technologies, and why here we have one of the most developed public and private systems in the matter. Thank you of all of you, and I hope you enjoyed the workshop. Thank you very much.
Thank you, Marcelo. So I will pass the floor to our friend, Ms. Alicia Barone, General Directorate of International Cooperation from Argentina at the Ministry of Foreign Affairs, International Trade and Worship. Alicia, you have the mic.
Thank you, Guillermo. So we have reached the end of this virtual conference. So in behalf of the General Directorate of International Cooperation of the Ministry of Foreign Affairs, International Trade and Worship of Argentina, I would like to thank the expert from INTA, CONICET, and the Ministry of Agriculture, Livestock and Fisheries for sharing our country experience on such an interesting subject, a key technology to improve food security and reduce hunger in the world. I would also like to thank Mr. Mohamed Ali Ben Abid, Ambassador of Tunisia in Argentina, Mr. Claudio Rosenweitz, Ambassador of Argentina in Tunisia, INTA and National Agronomic Institute of Tunisia experts involved in the implementation of the FO.AR project, development and biosecurity of drought-tolerant wheat. I would also want to thank the National Institute of Agricultural Research of Tunisia researchers. It is also a privilege to have the presence of so many specialized institutions from other African countries today. I would also like to acknowledge the joint work of the Argentinian Embassy in Tunisia and my team in the General Directorate in the organization of this webinar, and to all of you for being here today. As you know, the General Directorate coordinates the technical and scientific technological cooperation relations of Argentina with different countries all around the world. The Argentine cooperation actions are based on South-South cooperation principles, a broad framework of collaboration among developing countries that takes into account the interests and needs of partner countries. Through the Argentine Fund, the International Cooperation, FO.AR, Argentina has developed over the past decade more than 60 technical cooperation projects in Africa, promoting the exchange of knowledges, technologies, and best practices, always taking into account the African Development Agenda. In this time of crisis, under the particular circumstances caused by COVID-19, we have promoted a series of virtual activities within the framework of FO.AR projects, like this webinar. We strongly believe we must reinforce the role of South-South cooperation in promoting sustainable and inclusive development to reduce the impact of global threats to vulnerable populations. For this reason, we hope to continue working together to address the multiple challenges our countries face and to collaborate with the implementation of the 2030 and 2063 agendas for the benefits and well-being of our community. Thank you very much. Shukran.
Thank you, Alicia. Thank you, Marcelo. Thank you to the both ambassadors, Mr. Benavid and the Argentine ambassador in Tunisia. Thank you to our colleagues from INTA, CONICET, and from Tunisia. Thank you to be here today. So we are closing this seminar. I would like to mention that we have more than 100 people attending to the seminar from Tunisia, Algeria, Ethiopia, South Africa, Nigeria, Egypt, Mozambique, Rwanda, and a lot of countries, also from South America and Argentina. Thank you to authority, speakers, and audience for attending to the seminar. The whole seminar can be found at the YouTube channel of INTA please subscribe and follow us. We hope to see you soon in our next seminar. Have a nice day in Africa and Latin America. Thank you. See you soon.
Thank you.
Bye-bye.